Hi everyone, so this video can be used as either an overview or a summary of both cell respiration and photosynthesis, but not exactly the entire process of both metabolic pathways, but more so the big ideas of those pathways, and a little bit of comparing and contrasting between the two, kind of connecting everything together. So the first slide here we have is metabolism and membranes, or M&Ms, and the reason why I have put metabolism and membranes together is to highlight the importance of a membrane to the metabolic pathways. Okay? In fact, the membranes of particular energy converting organelles, such as the mitochondria in eukaryotes and the chloroplasts in the plant and the algae, the membranes are what are the most important because that's where the majority of the ATP is being produced. So when we think of metabolism, always think of membranes. Okay, so here we go. Prokaryotes use their plasma membranes to produce ATP, and eukaryotes use very specialized membranes. And those specialized membranes are inside what we call energy converting organelles to produce ATP. Okay, and so the mitochondria, or the mitochondrion would be singular, that's the membrane used in eukaryotes, and the chloroplasts, the membranes of the thylakoid and the chloroplast, would be used in plant and algae. Okay? So the most important characteristic of these two organelles, the mitochondria and the chloroplast, is that they both have very large amount of internal membranes. And that large amount of internal membranes is excellent because it increases the surface area. And if you increase the surface area of the membranes, you can have a lot of electron transport chains happening at the same time. The more electron transport chains happening at the same time, the more ATP that you can produce. So I always like to think of the phrase of lots of ETCs to produce lots of ATPs. Again, that's lots of ETCs to produce lots of ATPs. And don't forget that. The next slide here is showing you the process of oxidative phosphorylation in the inner mitochondrial membrane within eukaryotes. And that comprises of two parts. We have the electron transport chain and chemiosmosis. So here would be the electron transport chain, which was covered in the electron transport chain video, and then also this would be chemiosmosis, which was also covered in, not covered in the oxidative phosphorylation video. Okay, and so similarly though, in the chloroplast within the thylakoid membrane, we have, rather than oxidative phosphorylation, it's called photophosphorylation, and that's because we use light as our stimulus to provide the energy for the electron transport chain as well as the chemiosmotic process. Okay, so here's our thylakoid membrane. Light, we still have the electron transport chain moving, we still have the whole idea of chemiosmosis happening here, but there are some differences, right? So instead of oxygen as being a final electron acceptor, we are actually, and in order to make water, we're actually using water and breaking that down to get the electrons in order to make NADPH. So, the electron transport chain, electrons are carried from one site to another via carrier proteins and small molecules. So in the mitochondria, as a review, we have NAD plus that will take up 2 electrons plus the hydrogen ion to become NADH. And the NADH will shuttle the electrons from the cytoplasm to the inner mitochondrial membrane, right? So this is all happening in glycolysis. The electrons that are released then from the breakdown of foods such as glucose into CO2 are moved through the membrane by a chain of carrier proteins. The free energy released as those electrons flow down the path from a high energy state to a low energy state is what drives a series of 3 hydrogen ion pumps in the inner mitochondrial membrane. And then that third hydrogen ion pump is the one that actually catalyzes the transfer of electrons to oxygen, reducing it to form water. In the chloroplast, it's similar to this, but it's also different, right? So in the chloroplast membrane, or the thylakoid membrane, it consists of some important components that are not found in the mitochondria. So for example, photosystems are not found in the mitochondria. Photosystems are a type of carrier protein. And within the photosystems, we have a green pigment called chlorophyll, which is what actually captures the light energy and then harnesses it to drive the transfer of electrons. And then also the electron motive force in the chloroplast generated by the chloroplast photosystems drives the electron transfer in the opposite direction to that of the mitochondria. So electrons are actually taken from water to produce oxygen, and they are donated to carbon dioxide to make glucose. So therefore, the chloroplast generates carbon dioxide and glucose, whereas the mitochondria consumes them. And so here's another slide. You might remember this one from one of the previous videos on the mitochondrion. And then here it is next to the chloroplast. Okay, so as a review, remember everything in green are your inputs, and then the blue are the outputs. The flow of electrons is designated by the red arrows, and then the orange blobs here are your carrier proteins. Okay, so then chemiosmosis is again similar between the mitochondria and the chloroplast, but what I mean by that is you see the process in both the chloroplast and the mitochondria. So chemiosmosis is where energy stored in the proton gradient across the membrane is used to drive cell work, and more specifically used to drive ATP synthesis, but that's not the only thing that it can be used to drive. So chemiosmosis, if we break up that word, chemi means chemical bond forming reactions that generate ATP. Osmotic comes from the Greek word of osmos, meaning push. So osmotic refers to the actual membrane transport processes, or pushing something across the membrane. So chemiosmotic coupling, and here's a diagram of pictorially what I'm saying over here. The energy from either, because again we're talking about chloroplast and mitochondria, right? So the energy from either sunlight or from foods such as glucose is used to create an electrochemical proton gradient across the membrane, and then that gradient will serve as an energy store that can drive other energy requiring actions. So for example, it can drive membrane transport within the mitochondria and the chloroplast. It can also drive ATP synthesis in the mitochondria or chloroplast, or it can also even drive flagellar rotation or movement of bacterial cells within bacteria. So chemiosmosis occurs in two stages. Stage 1 is where we have high energy electrons that are derived from the oxidation of either glucose or sunlight, depending on if we're talking about respiration or photosynthesis. And then those high energy electrons are transferred along a series of electron carrier proteins that are embedded in the membrane, and that's what we call the electron transport chain. This transfer of electrons will release energy that is used to pump protons from water across the membrane, and then this generates what we call an electrochemical proton gradient. And remember that an ion gradient across a membrane is a form of stored energy that can be harnessed to do work when the ions are allowed to flow back across the membrane, and that's exactly what happens in chemiosmosis. So for stage 2, those hydrogen ions that we just talked about are able to flow back down the electrochemical gradient through an enzyme called ATP synthase. And then that ATP synthase, using the proton gradient, will catalyze the synthesis of ATP from ADP and an inorganic phosphate. This enzyme is an F-type ATP transporter. That means it's a turbine-like transporter, so it does work as a rotor, so to speak, and allowing the proton gradient to drive the production of ATP. And then here's the video, here's the picture of that. So stage 1, we have the electron transport drives the pumps that will pump the protons across the membrane, okay? And then in stage 2, when those hydrogen ions have to go back down the gradient, that proton gradient established will be harnessed by the enzyme ATP synthase in order to phosphorylate ADP and to make or generate ATP.